This next section, we're going to look at DNA and how does it code for the information. It's very important, and this section's a little long, but like I said, we're going to have lots of important information covered. So we're going to start looking at Watson and Crick and why their structure that they discovered for DNA is so important. So you can see them pictured here. And they knew at the time that the inheritance factors were kept on the DNA, but they didn't really know how that worked. And so these guys helped to determine the structure and how that worked. So if we look at the structure of DNA, deoxyribonucleic acid, we have several units, which are called nucleotides, that make up that twisted ladder. So each nucleotide is going to be composed of a phosphate group, a sugar, which is the oxyribose, and then a nitrogen base. The phosphate and the sugar are going to make the sides of our ladder, they alternate, and then the bases are going to pair up to make the steps of the ladder. So here you can see we've got deoxyribose versus ribose, and they're just different types of sugars. The main difference you can see here, this has a hydrogen and this one has an oxygen and hydrogen. And we'll talk about the difference of DNA and RNA in a little bit, but those are the main differences. So just different types of sugar. The bases are going to be consisting of four main types, and then there's a fifth one that we'll throw in there a little bit later. But you've got two specific types, purines, which are double rings. You can see there's one and two rings, and these are the purines. And so adenine and guanine are our double rings, and pyrimidines are going to be single rings. And that's cytosine and thymine. And later on, we'll talk about a third type, uracil. These bases are going to pair up so they make the ladder. And so we have to have one double ring match up with one single ring. The steps, or sometimes they call it the backbone of the ladder, is going to be made of your phosphate, sugar, phosphate, sugar. And then your base is going to come across and pair up with another base to make those steps. Chargaff made a discovery that told us that these bases pair up, and an adenine and a thymine are always going to pair up with each other, as well as cytosine and guanine. You will not have a G with an A or a G with a T. Chargaff's rules say that they have to pair up in this manner. And so you can see here, they will not fit together if they're not with their correct partner. Watson and Crick discovered that DNA looks like a twisted ladder with the sugar and the phosphates on the outside, the bases making up the rungs of the ladder, and those bases are held together with what are called hydrogen bonds. And so you can see that a little bit better here. So sugar, phosphate, C and G, and then these are going to be your hydrogen bonds, A and T, and again, hydrogen bonds. So how do we get mutations? And so you can see some examples of some mutations here. We're going to have to copy our DNA. And a lot of times when that DNA gets copied, maybe that's not done correctly. And so that's when we would get mutations. So first of all, we're going to look at how is that DNA getting copied. So we have our twisted ladder, and the ladder is going to be split, and we'll talk about how that works in a little bit. And then each half of the ladder is then going to get copied to make identical rungs of the ladder. each half is going to have what's called a complementary strand. So this side would be complemented by this side. But then we can make additional strands, two complementary strands, to match up with the existing strands. And so when we do this, we're going to be matching up those bases just like we did before. So here, A and T originally were together. When we split this apart, now we're going to put a T back with the A, and we're going to put an A with the T. And once those are complete, now we have two identical strands. When this happens, if those aren't copied just exactly right, we might get a mutation. And this is easy to happen because it's happening rather quickly. It's happening so many times, billions of times. And so sometimes it might cause a problem, other times it may not be that big a deal. We have to replicate the DNA as part of the process of sharing the information about what proteins to make. When that happens, first we have to use what's called helicase to unzip or split that original DNA to get the two that we're going to copy. Then we have what's called DNA polymerase that's going to come back and start adding in those base pairs to make the copies. It does check itself to make sure that it's copying correctly, because if it doesn't, that's when we have problems, mutations, and things that aren't going to allow the code to be read correctly. When we're looking at this code, it's going to be read in sets of three, which are called codons. So the proteins are going to be made from amino acids, and those amino acids are going to be determined by the sets of three. And there are 20 different amino acids that we could get from the sets of three. Each set of three is going to code for that amino acid, but specific sets of three are going to code for things like start. Where do we start reading this information? And some might code for stop reading here. So almost like chapters, when you're reading a book, and there's lots of words that continue on and on and on, or the start and stop codons, maybe you can think of a capital at the beginning of a sentence and a period at the end to tell you this is one set of information, like a sentence. Because you're not going to just continually read and read and read. You have to know where do a set of three start and where does another set stop. Once we have our sets of three, they're ultimately, like I said, going to code for proteins. And so maybe we have a set of three that is made of G, C, A, then that would code for this specific amino acid. And these are real hard to read because this is so small. But if it says maybe U, A, A, then that's going to tell us stop reading here. And then these codes eventually tell us what traits a organism would have. The proteins are going to be then put together by whatever amino acids are coded for in a sequence. One thing that is important to this process is RNA. So RNA is a copy of that DNA that then goes off and tells what amino acids and then proteins need to be made. So we're going to have what's called transcription, And transcription is going to be where the mRNA or messenger RNA is transcribing the code, it's reading the code of DNA, and then it's going to go and it's going to tell in the cytoplasm what amino acids need to be made. So it's translating. Now, the difference between RNA and DNA, there are a few, but the main difference is when we are making these codes, instead of A matching up with T, A is now going to match up with that U, that uracil that we talked about earlier. So adenine is going to get paired with U. Now, it's important that these sets of 3 or these codons are read correctly, and if they're read out of order, we could have a problem. So here you can see the red dog ate the cat. 3-letter words, all making a sentence that we understand. If we were to take off the T and then we read every 3 letters to make up a word, then we get her, ed, and then not even words. So it's important that we start reading at the correct space, or we will get jumble, and that's the same thing that our body tries to read or our cell tries to read. So jumbling can happen. And we could also make the wrong amino acids. So if we put this all together, we start off with DNA, sets of 3 bases, then those get read and transcribed into mRNA. So T gets transcribed into an A. A instead of T turns into a U, C, and G. T, A, T, A, and A, U. And so we read the pairs and decode them. And then the mRNA turns into tRNA, and we code them again. So remember, A instead of T turns into a U, U's an A, G and C, and so on, until we have that sequence coded. Then we take the sets of 3, and those are going to tell us what amino acids would be made. Then those amino acids go off and make the appropriate proteins.